So let's begin with the disease data. I showed that data, or those data, but I didn't quite understand what I showed. So what I take 3 different methods that predict the effect of variants, and I plug into it a data set of variants that cause disease, OMIM. Now, the 2 methods to the right here, polyphen and SIFT, so this uses alignment information, this just uses the blossom matrix, and this uses machine learning. Now, what the axis shows here is the ratio between effect and neutral. That means about round about 80% of this data set, so 80% of the variants that cause disease are predicted to have an effect. Again, for this method here, they have been used for training, so it's not cross-validation. I'm comparing slightly apples and oranges, but that is not my point here. My point is a different one. Let's just assume this would have been a completely cross-validated image, same data set, same cross-validation, no other ratio. I should talk about error. I'm not doing it at the moment, and the error is not the issue. My question to you is, does that mean, so I see a data set of things that I know cause disease, so I should predict that they have an effect, and I see that the ratio is highest here. Does that mean that that method is best? Again, this is not about error, standard error, it's just about the numerical height. I give you 3 values of 3 methods. I give you essentially for the set of OMIM variants, for the set of variants that I know cause disease, what fraction of those are predicted by that method to have an effect versus what fraction are predicted to be neutral. And again, for SNAP, that ratio is roughly, 79 or something like that. Here, this may be 85 or 83, and here it may be 78. Does that mean that this is the best? Again, it's not about cross-validation.
We don't have an error?
We don't have an error. I'm just asking, let's just assume, for simplicity, that the error bar is so small that you don't see it. This is a lie, but the question goes in a different direction. Yeah?
Is the data balanced, the underlying data between
No, the data is absolutely not balanced. It's just a data set of disease-causing things, and these are models that have been trained on something else. In fact, for SNAP, this is totally true. SNAP had never seen those data, had never seen the disease data.
How old are those classifiers?
So this is the oldest, the second, no, that's the oldest, the second oldest, and the third.
Okay.
All 3 of them, again, so they used some of it. So the story is slightly, it's SNAP 2. So SNAP 2, then this is the oldest, second, and the youngest.
And SNAP didn't use the data at all?
So again, the story is more complicated. On Tuesday, I told you that SNAP 2, one thing that we differed, we put this data in. But we have a version of SNAP 2 that does not use the data. That's the one we test here. So again, it's cross-validated for this one, not for this, but let's just ignore that reality. The catch is somewhere else.
Well, then I would say that you cannot say based on the height that this one is the best, because if this one uses the data, obviously
again, that is not the issue. So let's assume that they all have been cross-validated, that none of them would use the data. It's not reality. So I'm just not saying it's not true, but that is not the issue. Let's just assume that they all had not used those data. Okay, that's not the issue. What is the problem in this argument? So the argument is simply, it may be right or wrong that one method is better than the other, but the argument, the way I pitch it, is wrong. I repeat the argument. The argument is, this is the best method because for this new data set that no method had ever seen before, of effect variance, that has the highest score. So the highest score means the highest number of these variants are recognized as effect, which they should. Why is it still not the best? Why is there something wrong in this logic, something flawed in that logic? Did you find it? You're smiling.
Maybe. Well, I have an idea.
Yeah?
But you want
You can go first.
I guess because you're only looking at the positives.
Exactly.
Yeah.
Exactly. So this is a method that would always predict effect, would absolutely score best here. And we have to find a way to sort of measure or tease this out. This is not easy to do because typically you don't get neutral data. So one way of doing this, I'm looking at the training neutral. So I have a train set, the same train data set, and I ask what fraction in the training data set of neutrals is incorrectly predicted, so to speak. And so this one predicts many, many more neutrals. So in order to sort of make this point here that one method is better, you somehow have to measure it against this increase. So the amount that this is higher here than that, this is the amount that this should be higher than that. This, in fact, so visually this could be the case here. This is not my point in this particular case. My point is that just from one positive data set, that may be the only way you can get your hand on data, but to use that to say one method is better is just not good enough. So now we get into, I briefly alluded to that on the last slide on Tuesday, into the idea of not improving the method. There are many things wrong with the method, but rather look what we can benefit from the method. And one way we can benefit from the method is it predicts the effect of a particular variant. Since it can predict the effect of one particular variant, we can essentially predict the effect of every single one. We cannot predict the effect of coupled variants. So far what the method has trained only on is individual. So it has trained what's the effect if I turn this one, if I turn this one. But now I can simply in silico turn every single one. And I cannot only turn every single one against one particular variant, I can turn every single one against all 19 that are not observed in nature. So all 19 non-native variants. And I simply can predict the effect of that, and that gives me some sort of landscape of mutability or of resistance to mutability. Okay? Now the first thing I can do with that, I can ask, well, if I took every single residue and replaced it to, say, the letter A, what would be the effect? And then if I took every single one and replaced it to the letter C, what would be the effect of that one? And I can simply look, what's the average effect of all the 20? Right? So this is the kind of question that I will not ask for given your background in the examen, but just for curiosity. What would be the one that is sort of the most, or the least effect somehow? So I can, now what I'm going to do in the next slide, I'm going to compute the average over all 19 times length, right, for all the proteins that I have. I cannot remember what our data set is. Could be human, right? We look at all roughly 160 million variants that you could possibly have in human, and you get an average effect. Okay? And now you ask, at every single position there is a sort of effect that you get, right? The effect goes up and down. And now you ask, if I only, so that's the average effect of doing 1 against 19. Now the question is, if I chose one of these 19, if I always picked, say, the A, would that be representative of the average? Or would the C be more representative? Or the D, or the E, or whatever? Which of the 20 residues is most representative of the average? Does anybody know enough of the amino acids? Okay, so...
mentioned it once.
Well, then it's not important. Yes, I did mention it, but ultimately the one that is most, in some sense, most neutral is the alanine. It's a small amino acid. Again, you will not be asked that. People have historically always done in the lab alanine scans, meaning that they mutated particular residues to alanine, because they have been arguing for a long time that alanine would be the most neutral. And I found it very surprising in some sense. This is one of the few times in my work that I have come across something that everybody had always said. Usually when I looked with a machine afterwards, with a computer, without bias, there were typically what sort of were the general rules were always wrong. This one is actually right. So I very much like that one. Anyway, so you can then use that idea and try to predict hotspots. So now you can say, okay, I have binding sites, and I'm going to now look at particular positions in the protein where whatever I do, or has a particular effect, a strong effect. And then I believe those that are going to be affected by no matter what happens are the most important to maintain. Okay? And that sort of thing we can use to predict hotspots and protein interactions. But here is another visual example. No, this is not very visual. So this was supposed to... Okay, the movie is gone here too. This is supposed to... Just imagine it turns. Anyway, if it turned... I already pointed that out on Tuesday. What you would see is that there's redder around the binding site. Redder means more susceptible to change. Here you somehow get an idea that, again, there are three different ways of doing that. That is sort of the average of a non-native. This is you score things that are conserved more than others, and this is sort of a different scoring. This one correlates somehow, but there's a lot of red here. This correlates better. There is red in the binding site, but this binding site here is missed, for instance. And in this scoring here, both binding sites are picked up. And ultimately what this scoring is, it asks, is the variant, if I put that into a Blossom matrix, would the variant be okay? Now, if the variant is okay by biophysical constraints, by Blossom says it's neutral. Nevertheless, I predict an effect, so that must be something relevant. Okay? Because it's not a trivial reason why this variant cannot be mutated. Now, we can use that kind of logic and essentially estimate the sensitivity or the mutability in a particular protein. Here, that's a membrane protein. We had never used that before, and it worked very well. And actually, this is the kind of thing that also allows you to predict differences between two proteins, or between different susceptibility at different positions to the same identity. Let's just go back to that slide. So. What the coloring means here is the redder, the more this is sensitive to variation. And I can pick a site such as this one here. Green means it's fairly neutral. So we have two proteins that are very, very sequence similar. I can't remember, over 90% or something like that. And many residues are the same. But in this particular case here, no, that's not the case I want. I want a case where I have two different colors and the same amino acid. Okay, I'm not quickly finding. Well, here is sort of there. Where did you see a better one?
Last one, SS. Two different colors and same letter. Yeah.
Here, this is just, I got it. Yeah, exactly. So here we have, and there's an even more extreme one. So there's a switch between a red and a green. So what I wanted to point out is, again, you can predict a differential effect for a position that is identical. Okay, so it's not really, because this is ultimately the strength of machine learning. The machine learning is not picking up a feature that is an individual residue, but it's a feature that is coined by the environment. And the environment for these two sequences here is different. Okay, that is the unique idea. Okay, now ultimately, you may argue that every single possible SAF amino acid variant will be observed. Really? Do you believe? So again, let's just do the math again. What I briefly mentioned, we have 160 million possible SAFs in human, and I'm not entirely sure whether it's right. So the estimate goes as follows. We have 20,000 proteins, 20K proteins. Protein has about 400, human protein has about 400 residues. So essentially we have 20,000 times 400. So this is about 160,000, right? Roughly. 160K. SAF in human. Possible. I mean, in silico possible. So I can create 160,000 point variants in human, because I have about 160,000 residues in all the 20,000 human proteins.
8K, 800,000, right? 2 times 4.
Yes, times 20, or times 19. Times 19, and then I did 20. So I can do, yeah, that's a very good point. 8,000 is 160,000 minus 8,000 is 100. To be a little bit more precise, 152.
No, I mean, you have...
Yeah.
Okay.
You were right, I forgot this step with times 20 because every single position has 19 possible variants, right? That is exactly the factor I forgot. Thank you for making that point. And the total sum, I understand that you did that, but in the meantime I had enough time to work out the number a little bit better.
Yeah, sure. But what I meant is 20 times 4 would be 8.
Yeah, that was my mistake. I forgot. So it's 20,000 times 400 times 19.
I see.
And actually I put down the 20 and then...
I see.
Thank you very much for putting this. You're totally right. The numbers did not add up because I forgot the times 20. Thank you for being awake. So we have about 100, I still say we have about 160,000. Okay, now my question is, will every single one of those be observed? No, why not?
Because some are probably instantaneous death.
Exactly.
Yeah.
Well, whatever that means.
Yeah.
Instantaneous death. So most likely this cell will not develop. Yeah. So instantaneous death, I mean, it depends on the definition of that word, but most life will not happen. Yeah. Because there's a variety of knockouts that will be so crucial that the egg will not fertilize or the embryo will not grow. So life, it will never come to the point of being born. And then there will be some that are so fatal that you will die very early on. Again, there are these examples. But we currently, some people believe that everything else will be observed. So at this point of the number here, we have about ballpark hands-on.
Why is it K?
Wait a minute, there's something.
Why is it K?
Yeah, there's something wrong with it. Exactly. Thank you. We need a lock. I was just stumbling over the fact that I got the order wrong here. But why did I do that? 20K, yes, 20K times 400 is million. Yes, sure. Yes, thank you. Okay, simple math. And that is, in fact, the point I was just stumbling over. Observed at this point are about 10 million. So if there really would be 152, we are 1/15th of the way, so to speak. And these 10 million come from 65 million people. I will get back to that toward the end of today's lecture. But this is sort of the ballpark. And so some people believe that we will ultimately see them all. See them all, again, there's another thing with single nucleotide changes, you don't get all possible 19. So some are not SNF possible, so to speak. But in terms of mutability landscape, in terms of understanding where a protein is sort of most important, we can, in silico, do it all. And here's an example where we do that. So the sequence runs in that direction here. It's a long sequence. This one down here is cut out from that region here. Red means strong effect. Green means strongly predicted to be neutral. And we, in fact, so again, the sequence runs in this direction. What you have in this direction is the 20 amino acids. So we look at every single position. There is a white one, a black one. The black one is the native residue. So here in that position you can see it's an A, and then whatever it is, it's an H or something like that, okay? That's how you can read the sequence. Now, you see that there is a lot of red. So, but you also see that there are some regions where you have more green dominated, some regions, larger regions where you sort of have consistent red. Overall, you see a lot of red, but again, you see these green regions. Now, the question is, how much do I learn from that? And ultimately, what Max Hecht did with this here is he now looked at this particular mutability prediction and used some constraints. For instance, constraints about how does the family look? Constraints about where in the protein is that? So he essentially looked at all the red ones and asked, are they on the surface? So then he looked at the red ones that are on the surface, the most red ones that are on the surface, and then he looked sort of for connections, for contacts. Essentially, the reddest things that are in contact on the surface should somehow matter. And then he boiled it down with these kind of calculations or considerations. He boiled it down to just 4 residues, and these 4 residues are in fact vital binding sites for these 2 proteins. So with some additional information, 3D structure, some staring at this example, you can get from a mutability landscape that is largely red to something that makes a lot of sense. So this particular case is just to prove that it works, because we knew the binding site. But in principle, he could have done that without knowing that. But he could not have done that in the computer. So it's one way we can still not do. Even on the simplest level of per residue, so because of needing context, needing 3D structure, but in the simple residue per residue level, here's one particular protein for which, again, the sequence runs in this direction, and this one now, the y-axis, gives you the average prediction of all 19 possible variants, or the average effect. So things where this number is high here, they all have an effect. Essentially, all variants have an effect. Where this is low, only some variants have an effect. Where it's 0, no variant has any effect. So one way to see this experimental result here is that almost every variant has some effect. There are positions where whatever you do doesn't have an effect. And there are positions where essentially whatever you do does have an effect. Okay, you see everything. Down here, what you have is the prediction for the same protein. Okay, now how does it look? Is the prediction good? So one way of asking that here in a binary 1-0, if you essentially would say you get the state predicted right or not, how many belief do you have right here? Is it a lot? It's a lot. So somebody says a lot. Is that sort of, actually if you counted the ones and zeros, then you would look at 78%. So back again to the question, is that a lot? How do you answer that question? So 78 is not 100 and 78 is not 0. And 78 is closer to 100 than 0. Is that the answer? Or is there something else I have to look at?
Chance.
Chance. So 50% is random. So, well, you're clearly away from random. Now you would have to look at significance and things like that. What else could you do instead of the binary classification of classifying these 2 points or these 2 types of data or results?
Measure the difference. The closer you are at the smaller. So
essentially the correlation is I assume what you meant by that. And the correlation is 0.76. 0.76 is, in our field at least, a fairly high correlation. Anyway, so the exercise here was not that we can sort of predict in silico scans, or we can in silico predict experimental mutation scans. The idea was, the point was really to look at data, and the best way is to begin. So I've seen it best in Tobias, begin counting. So just have in your head create some marks over 50, over under 50 or something like that, and do a quick count. Get an immediate, because our visual impression about correlation is often misleading. So begin to count as soon as you can. Anyway, okay, now we get back to this issue of sequence variation in human. And this issue has to do, of course, with this question, how do I get from genotype to phenotype? So how do I get from the genome of a person to how that person may look different? So I have 2 different genomes from 2 different people, and they look different, or they have different phenotypes. So look is just one aspect of the phenotype. There are all kinds of other aspects that are not apparent, or not immediately visible. So how much can I, from the genotype, estimate the phenotype. That's one question. The other question that is relevant, of course, is how can I, from looking at the genotype, choose the drug to administer? So is there a drug that is sort of relevant for a particular genotype? So let's begin with the genome that is completely sequenced. In 2000, again, roughly 2000, there were two efforts here. One from a private company, Francis Collins was heading that from NIH. He then became the NIH director. Sorry, that's Francis Collins here. For those of you, that's Bill Clinton, who don't remember this guy. You may remember his wife better, more recently. That is Craig Venter. And Craig Venter headed the company approach. Now, so both of these do a human genome. What do they choose? And about size. So overall, this genome is 3 billion letters, so 3 billion nucleotides long, which translates to 20,000 proteins. In terms of information content, this is the same information content as in these 4,000 books. This is exactly 4,000 books. If you don't believe me, I counted them.
Did you print them, or how did you measure that?
No, I counted the books. Okay, and then there was some estimate about the. Yeah, so there is some fitting in here. Yeah. But the books are counted, and if you wonder how I can make sure that the books are counted, is a repetition of the same slide over and over again. I don't remember how. Well, you will. So I believe it's only this one here that is original, and then I duplicated them. Anyway, so. We still don't quite actually know how many proteins we really have. We talk about 20,000, and in fact this should be proteins here. The number of genes is higher because we have RNA that doesn't code for proteins, is also part of genes. So today people believe it's sort of in the ballpark between 30 and 40,000. And there's 100,000 constructs. 100,000 constructs means 100,000 different ways of sort of building these proteins. And we still don't quite know what the real answer is. But the number 20,000 proteins is one that we believe in at the moment. Now the next question then became, what's the diversity? How much change is there? How much do we differ? And one way to map the difference is any pair in this room, the similarity between you and your neighbor is the blue, and the difference between you and your neighbor is the red. One way to think about this is you have one difference in every single one of your proteins. I'm only talking about the proteins that change amino acids. Okay? The ones, if you want from genetic samples to find out that somebody is your brother or not, then you will use all variants, all nucleotide variants, and will include many, 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 many more. So because 99% of the genome is not coding or not amino acid variant, or it's not expressed as amino acids. So there are many, many, many more variants. So essentially you're looking at variants between you and your neighbor in the ballpark of 1 million. Because the 20,000 that I just talked about that it changes the amino acid is less than 1% of it. Anyway, so now the question is that a lot or not? Well, one thing that you have to remember is in this entire library, I have 35 trillion cells. And every one of these cells has exactly the same thing. Essentially there's no change. Right? My 35 trillion cells have exactly the same library to the letter. So in this context, 20,000 is actually a big number. It's much, much, much more than we thought. Now, the next question actually becomes, do these 20,000 between us differ? Well, you may argue, well, we look different, so somehow the difference comes from the genome. And again, there's always the big difference between men and women. So let's just look at the part where we have the same chromosomes. And so now the first question is, can we actually map it to known experiments, to knowing something about these variants? Actually, so I put this number out. If we want to wait until we can give an answer, we have to wait until 2050. Because at this point, for less than 0.1 per mil, do I have some idea about the place where this is, but not about this variant. I know something about the protein. That is as much as we know. And this, I believe, is absolutely not representative. So answering the question, do these 20,000 matter or not, we cannot fall back to experimental information, but we can use the computer. That's the method SNAP, developed by Jana initially, and then by Max. That predicts whether there's an effect or not. Now, what I didn't talk about so far, effect can mean two different things. Effect may mean that this protein breaks up, it no longer functions. Okay? Effect may also mean that the protein functions differently. So it gains a new function. It does something else. This is also an effect. The method that we have cannot distinguish these two. What we do know is we have looked at examples of very strong things that change function, and they have a very strong effect in our method. So we see that in exactly the same as something that is disruptive. Neutral is neutral, so that one is clear. And I'm going to show you a couple of slides of this type here that are cumulative values. Again, the score SNAP. Score 100 means very strongly predicted to have an effect, minus 100 very strongly predicted to be neutral, and cumulative means that I'm going to put a certain threshold at a number, say 75, and I'm asking my entire data set that I'm going to look at, right of that point, what fraction has an effect. So let's just look at if I had now my method, if I had a very good method and I started with a data set of effect in red here. And if the method would be very good, what it would predict, everything has an effect very strong. So that would be the predicted curve, right, of a very, very good method. If I started with a data set of neutrals, then almost everything would be here, right? That is so the ideal outcome. Just to look at 2 points. So 20 to 25% or whatever. Now this is reality. This is SNAP as what we believe is one of the best methods in the field. So when you look at this, so this is experimental effect, experimental neutral, and when you look at this, essentially the degree to which this one is not up here is the mistake in getting the effect variance, and the degree to which the green one is not here is the mistake in the green ones. Now, the degree to which it pulls these two things apart, that is the power of the method. Because if every variant would be predicted in between these two, then we would have random. Meaning a random method would not distinguish between effect and not. Okay, it would always on average predict the same, right? That would be exactly in between. So that is the entire power of the method. And if I now query that method with a data set of disease variants that have an effect, so again, what I have to say is my method here has learned molecular function. So we probed it most of the variants, about 80,000 variants that we had something to do with molecular function. Most of those were not from the system level. Now if I probe with something that is a system level OMIM, then this is what you see. And again, that is very remarkable because for us that was the first time that we came with a totally new data set. None of those had been used for training, testing, or whatever. They were not similar to anything that had been used. And nevertheless, the effect was stronger than experimental or cross-validation effect. Why is that surprising? So that question can be answered by people who have sort of some experience with machine learning applications. Yeah?
Usually when you train on a cross-validation set, you'd expect when you change to completely unseen data that the prediction is less or not so good for the new set.
Yes, I do not have your photo. So your name is? Konrad. So what Konrad says is absolutely true, and mind you, what Konrad says is true, although we assume that the entire purpose of cross-validation is making this statement that Konrad become wrong. Because the entire idea of cross-validation is that for your future data set you get exactly the same curve, right? But in most cases that fails. Almost always fails. Because we almost always somehow over-optimize. Completely right, Konrad. Nevertheless, in this particular case we are above. And it's not, I would argue, a lucky case. Why do you believe this could be the case? again, it's a suspicion. I cannot prove anything, all right? But my rationalization of this observation.
I guess it looks like as if there were a Latin distribution and the classifier learned that and now can predict based on that distribution independent of which data set it is.
No, that's true. Essentially, in a little bit of fewer words, you would say the machine learning generalized.
Well, yeah.
Right?
Yes.
But my question is, if it generalized, why is this data set higher than the other one? Again, as Conrad always says, there's always a tendency to sort of a little bit over-optimize, so the red one should actually be above. Because again, above means more effect for the entire data set, right? Yeah?
Maybe for disease mutants, the effect is very strong.
That is exactly what I suspect. So these effects are so strong. Some of these red ones here are, remember I said in the classification when I talked about the data that in some cases maybe that's a judgment call whether this color is red or whether you say that delta G of 2.5 is an effect of 5.5. So some of those are less strong than others. But these are really strong because every single variant causes a disease. That's a pretty radical effect, okay? And I assume that this is exactly what it is. That most likely there is something like overtraining, and some of that should actually be below, but then that is compensated for by the fact that they are on average so strong. Okay, now we're getting back to the question, the variance between you and your neighbor. And again, nobody for who we have this variance, we start with 10 million here, nobody was sick, so sick that they couldn't be sequenced. So one definition of healthy is you get to a certain age, the age you reach means already that you have a certain level of health in terms of your genetic makeup. You haven't died so far from any major problem, right? And some of these SAFs will not be observed because they will never be born. But some of them will lead to situations where you die before the age that you have reached. In fact, there are many children diseases, such as progeria, where you, at the age of 16, look like 85 or 90, and I don't believe anybody has survived 20. So there are diseases such as this one. There are several of these diseases, many of these diseases actually around. So the fact that we can have the variant already means that this is a healthy person. Back again to the question. You and your neighbor, where do you believe in this curve here will the curve fall? So I'm going to now plot, not quite for the 10 million because we could not get sequence data for all of them, but for 7.5. I'm going to give you the answer for 7.5 million. I'm not entirely sure whether it's the recent 70.5 million data set or some other data set, but they are close enough. Where will it go? So again, it's of course not plotting the mutants between you and your neighbor. I haven't done a sequencing for you guys. It's between, for all 7.5 million, for 65 million people. Where will it go? Where do you expect it? Can I have somebody step up and say something and then argue for it? Who cares whether you're right or wrong? The important thing is how you argue.
Well, I have the suspicion that it doesn't make that much of a difference. There should be definitely an effect, so it should be in the upper right triangle, but I guess it should be under the experimental effect.
So the experimental effect are typically things that are disruptive. If every single one of your 20,000 variants would be disruptive.
Yeah, okay, it should be well.
So I do not believe we would all walk in here happily.
Yeah, I see.
But again, so this is, again, every single one of these here, almost every single one of those, causes a disease that makes people not walk into this room. So that's one way of thinking about it, okay? So another way of thinking about it, a substantial fraction of those variants here you have never seen. because they have severe issues with their lives. Or when you see them, you totally see that. This is not what we have here. So we will clearly not be somewhere here. This is clear. For me, the very surprising reality was I had assumed that we are going to be very close to here. And then there is some deviation from here, which is the effect that makes us different. In reality, this is what we saw. The curve is so thick, not because of error estimates. In fact, you do see, for these curves, you do see the error estimate. Okay, you see them as an arrow going up and down, but they are so small, these arrows, that the lines are thicker than the arrow at each single point. So it's tiny, tiny errors because millions of data points. That's why the error estimates are extremely small. So the line is so thick because of historical reasons. Because the first person who did it put it that thick to mark the point that it's there. Okay, that is all. It has no other meaning. And we were very surprised, in many ways this is one way of looking at it, for instance. So SNAP has an effect up here. We know the effect is stronger here than here. We cannot say that everything here is an effect because we somehow make prediction mistakes, but we know that if we say pick up something like 50, that clearly is an effect. And this is for 25% of all the 7.5 million. We have something that is in the ballpark of 50, that it means strong effect. And that is one way of saying this is surprising. So while I sort of tore your argument down in some sense, saying they cannot all be effect, a substantial fraction is. And that clearly is surprising. Okay, now we get to the next question. Okay, if there's so much variation between us, how is it actually between us and the Neanderthal? I'm not going to show you Neanderthal, I'm going to show you Homo denisovans, because at the point that we did this slide, there was not the complete Neanderthal sequence, only the Denisovan data. So where will that fall? Where will that fall? It's not shown. I wait for some input before the next slide. I'm sorry, it's not the curve you can't see. It's the curve on the next slide that you will see. And the curve will be very choppy because we have only one individual for that. So the error estimates would be very high. And you immediately see that in the choppiness of the curve. So the surprising thing is, it is toward neutral. All right? If that is with Denisovan, what about the gorilla? Between us and the gorilla. So if I only looked at, I have to admit, so as long as we talked about variance in human, we have apples and apples. When we talk about human and Denisovan, and when we talk about human and gorilla more so, we talk apples and oranges because it's not exactly the same proteins anymore. So gorilla has a different set of proteins. They have in the ballpark of 20,000, but there are some differences now. So all we can do now is look at the subset, and I cannot remember how many those were. At the point, I believe 16,000 that we did this slide. For 16,000, we could establish that the human and the gorilla were, overall, we could align them, so we call them homologs. Homology-based inference, the technology I talked about, would be applicable to this pair. And then we asked, for all the variants where they differed, what would be the predicted SNAP effect? So essentially we sort of saw gorilla as a replacement from human. Okay? And we did it one by one. We didn't look at all of them, that's impossible. So by one, and then we simply summed over the effect. Okay? And we did the same with the Denisovan, by the way. Now we have, interestingly enough, another sort of flip side of machine learning. So we surpass, again, a cross-validation, this time the green one. So we could now retrain on the blue being the new, and in fact, since we observed that, we have been discussing it, still haven't done it. But this is something that we have been wanting to do for a few years now. If we go further down in evolution, so we get more and more to different fruits, so to speak, because fewer proteins overlap, mouse, but it gets actually toward more neutral. And so what I don't show here, if we go to fly, it's even further down. And that's why we stopped. Again, now we have some of these variants that matter, and one way of looking at it is clearly our genetic differences do matter. There's no doubt. Some of those actually are even as high as these OMIM variant. But then the argument that some people would have, okay, those that make a difference, they do not happen frequently. Okay, there are two different types of variants, or actually there are three different types of variants. There are variants that we will call rare and frequent. Rare means less than 1% of the population has that. Okay? Frequent, more than 5%. Everything in between these two we throw out. We say we just want to make very clear story. So variants that are not very frequent, again 1% may be a high number, and ultimately we will have enough data to drop this and make it even smaller. At this point, less than 1% of the population is considered a rare variant. More than 5% is considered a frequent variant. One way to think about a frequent variant is those are variants that are true for a subpopulation, some local area or something like that. Okay, so what we expect is that the rare variants have more of an effect. So they are the kinds of things, let's say evolution tries out something. And if it doesn't work, it's only one small part of the population, but it's not put into the population, it's not reproduced because that did not really have an effect. While the ones that are sort of reproduced more often are things that have less of an effect. That's what we would expect. And that, of course, is not true. It's just the other way around. So random is up here, rare is red, common is green. Common is above, green is above red. So common variants that are shared by more than 5% of the population have more effect on average, predicted, than rare variants that are shared by fewer than 1% of the population, which again is a complete surprise. What does it mean? Since it is a big surprise, we don't quite know. Now, it clearly is true, so this is one paper that Jana Bromberg published in PNAS, in which she makes the point that there are a bunch of mildly, almost neutral, but slight effect sum up to effects that make up our individuality. That could be behind that something, behind that even. But let's go back to another view, and I talked about that already. The sickle cell anemia variant, E6-V, causes a bad disease, but the same bad mutation increases the resistance against malaria. So maybe this is the sort of thing. Maybe there are some variants that under some circumstances they would be good to survive, and ultimately I don't have to reproduce that. So when I look at the ones that have an effect, wherever I put the threshold, maybe I put the threshold here or here, I say there some of those have a bad effect and some have a good effect. The ratio here is only for visual reasons. I don't know what the ratio is. And maybe that explains why they sum up to something so big. So maybe more have a good effect. I do not know how to measure that, how we can distinguish, other than really making experiments. Again, we have experiments only for less than 0.1 per mil. So we cannot, it will take at least 50 years before we have this experimentally. So again, I have no idea whether it's half-half, I have no idea how it really is, but all I want to indicate here is some of them are beneficial, some of them are not. Okay, let's get back to personalized medicine. So personalized medicine somehow is the idea. One way of formulating the goal of personalized medicine is there's got to be a drug for each person. Is that realistic? Why not?
There's so many people.
There's so many people. That's one way of looking at it. So let's just be a little bit sort of less friendly to so many people. What about 1% of the people? Could we do that for 1% of the people?
For the richest percent, probably.
Okay, so that brings us to the next question to you. On the next slide, I'm going to show you time and number of new drugs added to the FDA, the Federal Drug Agency in the US, but it essentially is the same for Europe. The number of new drugs in a year. How many new drugs are added to the list of drugs we have in a given year? What do you believe? Ballpark figure. I don't care about plus minus some number.
1,000.
1,000. Who believes it's more?
Just approved or new drugs on
That's pretty much the same, because non-approved drugs are typically not handled.
Okay.
Certainly, so they are typically in this kind of thing that handles the kind of thing that the police is after you then.
No, that's not what I meant, because there's like a process to approve a drug, right?
We get to that. We get to that point. No, really on the market. But ultimately this will average out.
Yeah.
So 1,000 is the only number I've heard so far is 1,000.
Tens of thousands?
Several thousands.
Yeah.
Some
tens of thousands, like.
Tens of thousands.
Yeah.
Okay, that's so. There's some doubt where you falling if you have a doubt in tens of thousands.
It's 500.
So this is below. Here's reality. This is not K.
That's 40.
This is really 440. Stop. So no M, no K, no nothing, right? And you do see, this is, again, it's not the most recent one, but it's not that, it has not changed that much. You see that there are years where it's less than half of this 40. So it's very, very, very few. And part of the reason why it's so few are in these two numbers. On average, it takes about, and this number is up now, $2 billion to develop a drug. Okay, now we get back to what Tobias said. The richest people in the world, they could possibly, if they had a disease that is so bad, they could pay the $2 billion. Yes, but there's another number.
Yeah.
It takes 14 years on average. Again, some drugs take longer and some drugs take less. But on average it takes 14 years. So that means if you are the richest people, one of these people who can afford the $2 billion, you most likely will not have a disease with which you can just sit there for 14 years and wait for it, right? So even for the very, very, very few, this is not the scenario. Personalized health has nothing to do with developing a drug for a particular person. Yeah?
But why does it take that long?
That ultimately it is it's very difficult to answer your question for me. So there's a bunch of reasons. Let's begin with, we do not understand the body well enough. Most proteins in human, we do not exactly know what they do, okay? We cannot predict protein structure. In order to see exactly how a particular drug affects a protein, you would have to be able to completely compute how the binding of the drug to that protein changes. That means, first of all, you need to know all 3D structures of all the proteins. You need to know, you need to be able to do the computing, and then you need to be able to understand the complete chemical space in a way in which you know that this particular chemical would fit in there. And all that we cannot do. We don't understand the chemistry enough, we don't understand the molecular dynamics well enough, we don't understand the protein enough. We don't even know which protein to take. Okay, so this is a lot of lack of knowledge. The next problem is that the complexity, every single drug is targeting essentially a single protein. Okay, but now in many, many cases, adverse drug reaction, ADR, is exactly the problem, that you have reactions from taking a drug that are not the wanted ones. Because it's not only that one particular protein, it's in an organism with many different types of cells. That complicates the matter even further. So typically the way drugs are developed is you spend the first years on getting something that fits exactly to the kind of disease that you have, and that then that makes take a year or a little bit more than that. Then you make this drug better and better and better and better. That makes another 1 to 2 years. And then you have something that binds very tightly and has all kinds of ADRs, all kinds of side effects. So then you begin to make it less. Then you have to have something that people can digest easily. So that is not big masses. We're not going to have a kilo of drug every day. We have to eat it such that it has to be something that we can actually put into our body very easily without being injected. So there comes a lot of additional constraints. So you get from something that binds very well to something that binds much, much less well, but is compatible with all these environmental constraints. And then we still have something that we in principle in the molecular biology we know, but then we have to test it in humans. And then, so the most of that actually is the test in people, a substantial fraction of that, where you then the first round of testing is it actually doing anything bad to a person? That's the first round. And some drugs fall out in that round. And then in the next round, is it doing exactly what it's supposed to do? So is it actually helping in that disease? And all of this takes many, many, many years. Yeah?
How about tailoring a drug to the person?
That's what people do today. Oh, tailor, what do you mean tailoring?
Yeah, like adjusting the drug so it suits that person perfectly.
Yeah, but that is... oh, wait a minute. So again, what do you mean by tailoring a drug? Every single... We have 40 new drugs on the market. And now you want to tailor a drug to a person, or what do you mean?
I know that for example, for anti-allergens, there is a therapy which is tailored to the specific allergens.
So now the story gets different. So this is the story of cocktails. And ultimately the story of cocktails is partially because the numbers are not 1,000, not 10,000. Because we do not have that many drugs, and because they are so expensive, the only way you can sort of that's, by the way, also in terms of... Yeah, I'm online. In terms of economy, that's why pharmaceutical companies are so large. Because it's an immense investment, and again, this is an average. So some drugs are going to be 14 years in this pipeline and will not bring anything. And once they bring something, they bring it only for 26 years. So there are a few blockbuster, so-called blockbuster drugs that essentially pay for all the developments of things that don't work. And only very big companies ultimately can do that. That's why drug companies get bigger and bigger. It's not necessarily a result of capitalism in a pure world there. It's part of really the forces, let's call it the forces of the market. And you see the forces of the market in these few numbers here. And cocktails ultimately is the way to sort of connect or have a mix of different drugs. This is how we sort of attack HIV. This is how antibodies are attacked, so allergens are attacked, because there you essentially can combine things that are already approved. Now we get to the next, by the way, on the next issue. So when you take your drug, you get this long piece of paper that says all the things that could happen to you if you take the drug, right? Now this is ultimately, again, part of these 14 years. Then you have this entire list. Okay? In many cases, when you see somebody who is diseased, they're not taking only that drug. They're taking this drug too. And these come both with these long lists, right? How do they interact? Nothing is known about that. Absolutely nothing. So people now on a level of molecular experiments give two drugs. And there is a lot of the modern data collection in that direction. But on the level of the organism, the only running test is essentially the people who take drugs. And it's not very well recorded. So the data, so from that is not easily available. So some people, for instance, use some stuff of Google to get ideas about adverse drug reactions by combining two drugs, simply from social nets. That's one way in which people try to get some idea what exists. Anyway, so ultimately, this is exactly the point. So personalized health is not a particular drug. It's about cocktails, which ultimately is choice. It's about diagnosis, and it's about food. It's about things, the way you do your lifestyle. This is what is going to be affected in the near future, or is being affected already at the moment for some diseases. If you have one of these rare diseases, of these OMIM variant caused diseases, your genome in many cases will already be done if you go to Rechtsdeisa, for instance, and certain things will already be scanned. And there will be more of that. And ultimately, it will lead to things such as these drugs you talk about, and it will lead to choosing a different drug, because we know if you have high blood pressure, there is a particular drug that for people who have a high blood pressure is fatal if they have a particular genotype. We know that already. Again, this is whoever gets that drug is being sequenced, but there will be more and more and more of these cases. But ultimately, that means that in some sense, the precision medicine will remove the bad effect. So we'll somehow turn the good effect into something that is a bad. We will control by a different lifestyle, by different drug cocktails. We will somehow quench the bad effects. We have few of these bad effects because we cannot take. Well, ultimately, maybe we will be able to edit our genome, but at least in the near future being 20 years, the next 20 years or something like that, most likely we will not have a large gene editing in place. Because we again don't understand it well enough. So most likely this will happen. So we will have a little bit of reduction of that through quenching. So ultimately what it means, our genetic differences do matter for much, much more than just we look different. Okay? And ultimately they make us be individual and they make us have problems. These variants are not good for us. It's not what we always wanted, to be an individual. These variants are good for the species. So if conditions changed, and if that particular variant is an advantage, that will survive. Okay? What do I care as long as the conditions don't change? But that's bad luck for me. I'm part of the species. I don't survive as an individual. I survive as a species. So it's not really good for us. The diversity is good for the species, not really for me. And in some sense for me, by sort of reducing this bad effect, we can, by personalized medicine, by understanding our genome, we can somehow combine the best of both worlds. I do something that's good for me because I reduce the negative effects of particular variants, and we still, as a species, benefit from the diversity. Now, ultimately, this needs, underneath, we need some molecular understanding of a disease, and currently where we are is, say the goal would be to distinguish between a lemon, and in English a lemon is a car that doesn't work very well, and an excellent car. Okay? I'm very biased. I love that car. In 2 years it departed from me. It was too old. Anyway, now, so one way in which we can do that is we simply can count how many repair hours to have. And when you sort of look at a feature that correlates to fewer repair hours, then that is color. White cars are less often in a garage. Now that has nothing to do with them being less lemons than that. The reason why white cars is simply because we can see them differently. This has nothing to do with the quality of the car, right? And this is the level that we have currently reached with diseases. So we can say that a white car is not going to the garage more often. The white car goes to the garage less often, which helps you zero for the real problem of fighting the disease. That is currently where we are in terms of using the genome. Where we have to get is to really sort of understand the molecular detail on various aspects, refine many things, and then we can learn from it. And I believe there are many efforts being put together at the moment worldwide. And I believe it will be a long-term effort. So it's not the kind of thing where we will have something tomorrow. We will have some things tomorrow, but not that long. It all went into a TEDx talk, an earlier version of this, which put my life sort of upside down. That was the idea. If you haven't seen TEDx talks, just look at TEDx talks, make it part of your life. Understanding the science and the world around you is something that is worth it. TEDx Munich, or TEDx TUM, by the way, is an annual event that happens in December. Very much recommend. Now it's a full day to participate. It's fun. Thanks a lot to many of the people behind this work, the group, and here's an afterthought. So for this afterthought, I believe